Hello, good evening everyone. Mrs. Burr here, and what I'm going to do today is I'm going to take you through the first of two major functions of DNA, and this is going to be the start of a series of 4 sessions about DNA and protein synthesis and mitosis and meiosis. Okay, so let's look at what we are going to cover this evening for the WJ specification. Now, the first thing is, can you explain the semi-conservative replication of DNA, including the roles of DNA polymerase and DNA helicase enzymes, and can you use the evidence from the Meselson-Stahl experiments to actually show that DNA does actually replicate in a semi-conservative fashion? And then are you able to draw up some diagrams of your own so that you can explain it for yourself? Okay, so the first thing I think we should do is we should look back at DNA. Now, I'm sure you're very knowledgeable about DNA. Lots of students really like DNA as a topic and love learning about it. And what I'm going to ask you to do is this is the first of the pauses I'm going to talk about. I'm going to ask you please to think now. There's plenty of stimulus material on that slide there. Can you think of a list of key terms that if you were asked to give, let's just say you had a 6-marker on DNA in an exam, and you wanted to get all the key bits of information down about DNA, what sort of words would you actually put there? And as I say, there's stimulus there. If you've got time, you can write it down, but if not, don't worry. So I'm just going to pause while you think. Right, so as I said, the pauses are going to be quite short, so let's have a little think. Now, I think quite a number of you might have actually thought, oh, isn't that all about the double helix? Because essentially, obviously, that is certainly correct. And that actually you might have thought, oh yeah, A, T, G, C. But remember, at A level, we need to get used to this idea that we really should be naming them properly: adenine, thymine, guanine, cytosine. So great, if you've got that, fantastic. DNA actually has a sugar in there. Now, you may remember that it's a 5-carbon sugar, also known as a pentose, and the sugar's name is deoxyribose. So brilliant if you'd thought of that. Next one, part of the DNA structure has organic or nitrogenous bases. Sorry, that means bases that contain nitrogen. So that might be something else that you've thought about. And actually, if we're thinking about bases, it might have come to mind that actually those bases are held together with hydrogen bonds. Now, because those bases are held together, they're held together in a complementary fashion. We call it complementary base pairing. So you might have thought, oh yes, A with T, G with C, adenine with thymine, guanine with cytosine. And the DNA molecule is made of a series of these building blocks, monomers. So I'm sure when you've done proteins, you've talked about amino acid monomers, and when you've done lipids, you've talked about fatty acids and glycerol, and so on. And with DNA, what we've got are nucleotide monomers, and they actually build together to form DNA, which has 2 polynucleotide chains. So moving on. Now, this is sort of something that you may have thought of. If you have, diound, fantastic, because this is brilliant. And actually those bases are actually split into 2 types of groups. They're purines or pyrimidines, and actually T and C are the pyrimidines, and they're a single ring structure, and A and G are purines, and they're a double ring structure. Now, I've got a very, very silly way of remembering this, and this has sort of stayed with me for years and years, but I look at TC pyrimidines and I think I'm going to see TC, the pyramids. And then I always think, oh, those pyramids are huge structures, so that must mean the opposite. They've got to be the single ring, not the double ring. I know it's strange, but that's something I always do. Okay, so moving on from there. The other thing you might have thought about is the sugar phosphate backbone, because obviously alternating in the two parts of the backbone there's sugar, as we've mentioned, and also a phosphate group. Now, you might have also said, Dione, again, if you've thought of this, that those chains, that backbone, actually are built in an anti-parallel fashion, and that just means that one is upside down compared to the others. And you may also know in that case that actually the end of the chain may have a 3' or a 5' end, and that's nothing to sort of get concerned about, nothing to worry about. It's a dead simple thing to remember, and that is that it just means which carbon atom, so it's carbon 3 or carbon 5, is attached to the phosphate group. So that's a real quick recap of DNA structure, so well done. What we're going to do now is we're going to look at a past paper question. So just a quick little check now for ourselves. Now what you can see here is a question which basically says nucleotides are the building blocks of nucleic acids. So we're talking nucleic acids. They have an important role in many biochemical reactions in cell metabolism and regulation, including transport across cell membranes. The diagram below shows a nucleotide, so we know this is one of the monomers. And then it says identify components A, B and C, and with reference to components A and B, shown above, describe the composition of the nucleotides. Okay, so the first thing I would say to you is just be really, really careful with this question. I'm going to give you another short pause to try and think about the answers, but this question asks you about nucleotides. It doesn't actually say which nucleic acid. So careful, as I've said there, it doesn't tell you which nucleic acid, and that is really important with part A1. So have a go then. What are components A, B and C? If this was not any nucleic acid in particular, what could that be? Okay, so now we've had a little go with that. Let's have a little self check. So Dione, if you've got phosphate, and the second one, 5-carbon sugar, pentose. Now, do you notice there, I've not said deoxyribose, or I've not said ribose either, because it hasn't asked you about the components of DNA or RNA. It's just said a nucleic acid. And then the last one, nitrogenous base or organic base. And then the last part says with reference to A and B, so just A and B this time, what's the composition of the nucleotides of DNA, RNA and ATP? Well, if we're talking about DNA, as we've just talked about, A is going to be a phosphate group, but it's going to be one phosphate group, and B is going to be deoxyribose. That's the name of the sugar there. So now we're differentiating a little bit between what B actually is. Whereas in RNA, it's very, very similar, but this time the 5-carbon sugar, slightly different, it's ribose. And this is the nucleic acid that people always forget about. So this is just something, note to self here really. If this is ATP, then A represents 3 phosphate groups, and the 5-carbon sugar in this case is definitely ribose. Okay, so there's our little recap of DNA and some information about nucleic acids. Let's move on a little bit now and let's focus on DNA itself, because what are the possible ways that a copy of DNA could be made? Now, there are 3 potential suggestions there that I've got for you. And if we look on the left-hand side, if you have a look at me with my pointer there, you can see the original strand of DNA I'm talking about. Okay, so here we go, we've got our double strand there. And there are 3 potential methods in which that DNA molecule can make a copy of itself. So the first method, the top here, you can see the original DNA, still 2 black strands there. And what happens in conservative replication is that a completely new double-stranded molecule of DNA gets made or gets synthesized alongside the original. Okay, so we end up, after 1 generation of this DNA replicating, we've got a whole original molecule of DNA, and then we've got a whole new molecule of DNA, we'll call it. In semi-conservative, what's happened is the original molecule of DNA has actually split, and the new DNA has actually been synthesized alongside it. okay, and that is semi-conservative. So in the first generation after the molecule has undergone replication, what happens is that you end up again with 2 molecules of DNA, but this time each molecule is half original and half new. Okay, so half the original template and half new. And then the third method is dispersive replication. And in this case, it's a little bit more of a sort of a fragmented approach, a little bit more patchy, I suppose I'd call it, in appearance. You can also see from the diagrams on the right-hand side, again, this idea of how they'd look like. So we now know, don't we, that the semi-conservative replication of DNA, right? That is what we know is the actual mode of action. So what we're going to do now is we're going to look a little bit at how it is that actually is the case and how it works. Before we go any further, we need to look, because if you remember right at the start, those learning intentions showed us actually that there were 2 enzymes that we really need to know about, and they're DNA helicase and DNA polymerase. Now, we haven't got time here to recap the enzymes topic that I'm sure you've covered before Christmas, but in any case, just remember, and I've shown you some images there, the enzymes work by that induced fit method. They actually work in that complementary fashion between the enzyme active site and the substrate, and in this case it's the DNA molecule, and actually what they form is an enzyme substrate complex, and you can see that formed in the image in front of you. Okay, so enzymes, you know, let's think about the big picture. The big picture is enzymes play such a pivotal role in metabolism, don't they, that actually you've really got to know your enzyme work in just about any question you'll come across. So moving on then. So the enzymes there, so the helicase will actually break the hydrogen bonds between the complementary bases, okay, in the specific region of the DNA molecule, and the 2 strands there will separate and unwind, and the nucleotide bases are actually exposed. Okay, now what you can see there, they're shown in different colours for just for sort of ease of your explanation, really. You can see there that we've got the template, all right, they've called them both the template, but you can see they're in different colours just so you can see how the bases actually will actually form together in a complementary fashion. So what happens? Well, basically the helicase will unwind the DNA molecule, and those complementary bases will become exposed, as hydrogen bonds will have broken, and then along comes the polymerase. And what the polymerase does is that it actually, when the complementary bases, you can see them here, T with A, G with C, when these come in to the template strand, what happens is the polymerase will actually form a covalent bond between the phosphate sugar backbone and sort of join them together. And actually what happens at the end of all of this is that you may have several fragments of the backbone, and along comes another enzyme called DNA ligase. I thought I might as well just mention that. Along it'll come and it'll put all of the fragments together. That's just another little image. You might find that a little easier to see. You can see the template and the new strands there, but it's exactly the same thing that I've just shown you. Anyway, okay, so we're just going to pause a little bit again, because now what we're going to do is we're going to do a true or a false. And what I'm going to ask you to do is I'm going to ask you to think, is this true or false? So does DNA helicase break the hydrogen bonds between complementary bases in a specific region of DNA? True or false? So you can either say this to yourself or you can actually write it down. Second one, DNA polymerase. This forms the hydrogen bond between the 5-carbon sugar deoxyribose and the phosphate group. True or false? The backbone of the DNA molecule contains the bases with nitrogen atoms. True or false? Number 4, A is complementary to T and C is complementary to G. True or false? And then number 5, each DNA template produces 2 new strands. So, okay, here we go. True or false? First one, yes, absolutely. The helicase, as I say, breaking those bonds, unwinds the DNA, and then the polymerase can get to work. So the polymerase, no, it doesn't form the hydrogen bond, does it? It forms the covalent bond, so that was false. And then the backbone contains the bases. No, you know that, I'm pretty certain. The backbone is formed by alternating sugar phosphate groups, so well done, Dione. Number 4, this is a bit of a trick one, this was. I deliberately put a spelling mistake in there for you because this is something that lots of people do. They spell complementary as if they're giving somebody a compliment, and it's not. It means complementary, the way they fit together. So sorry about that, but that was false. And number 5, yeah, that's right. The template forms 2 new strands of DNA. So well done for having a go at that. So let's have a look now at a past paper question. OK, and what we've got in front of us is a typical question on this topic. And it says the diagram below illustrates replication of DNA in cells. And you can see there, just as we've looked, you can see there's a template strand, and what's happening is that along there you can see that the new strand, new molecules are being synthesized, okay, along the new strand alongside the template strand. And actually what I will say to you at this point is something I say to my own students: the best way to learn what's happening here is to draw it out for yourself. You just think of an order of bases, you know, knock yourself out, have a little look. What are the order of the bases? You decide, and then draw out 2 identical strands. And actually then you don't need a biologist to check. You can just check with somebody at home. Have I actually now drawn 2 identical strands from it? OK, so you know, that is the sort of, you know, piece of advice about learning DNA replication and what's going on. So let's look a little bit more at the questions here. OK, so the first question actually said, if I just go back to it, sorry, it actually said describe the sequence of events shown within the dotted rectangle in the diagram above. So the first thing to remember is that it's within the dotted rectangle, okay, and that is something very easy not to have noticed when you're actually answering these questions. I'm having a little go again about the word complementary there. And then the first question says describe, A1, sorry, says describe the events in the rectangle. So what we're going to do now is we're going to roll out our knowledge about DNA helicase. We're going to say it breaks those hydrogen bonds between complementary bases. You can give an example: guanine and cytosine, thymine and adenine, and then you can say then that the DNA will actually unwind. So you've described what's happening there, and you can see that it's unwound. There are complementary bases. Some of them are coming in. You know, you can actually talk about that then. The next question asks about the polymerase, what the role of the DNA polymerase is. And you can actually see here that actually I've said, well, that's forming the covalent bond between the sugar and that which is deoxyribose and the phosphate group, and that's forming the backbone. And again, I'm putting sort of some detail in there on my answer. Next thing, why is it semi-conservative? You know, this is quite a common question, and you need to have your answer ready to go on something like this, okay? So what you have to remember is the DNA molecules that are formed in replication. You've got your original template strand here, and that is acting as a template to form the new strands alongside. So semi-conservative replication is where we've got each new strand has been made of half of the original template strand and 1 new made in replication. So that is the sort of question you are likely to come across as far as DNA replication is concerned. So now let's move on to the most beautiful experiment in biology, perhaps, because this is what people called it when they first heard about it in the 1950s. And you might be thinking, what, Meselson-Stahl, the most beautiful experiment? What about Mendel? What about Mendel and his experiments with tall and dwarf peas? What about Watson and Crick in 1953 and their model for the structure of DNA? But actually, yes, people actually said this was the most beautiful experiment because they said the DNA was a model, and what Meselson-Stahl did was they set about, and they did prove that that model was correct, and the way in which it replicated was semi-conservative. Okay, so they actually set about to prove it. And there's a photo there of them back in the day and also very recently. So what did they do then? Well, they set up a series of experiments and they were called density gradient ultracentrifugation. Quite a long word. And actually what you can see here, if I just get my pointer out again, is that they were putting samples of DNA in a tube. Now they grew E. coli, Escherichia coli bacteria, and actually ideal choice because they grow quite quickly and actually they've got DNA in them. So, you know, good choice of organism to use. And what they did was, I'll talk about the samples later, but each of the samples that they produced were put in the centrifuge, and it has a rotor and it spins round, as I'm sure you know. And actually what happens is that the dense parts of the mixture there will be thrown to the bottom end of the tube here, you can see, and the lighter parts won't be thrown as far, they'll be further up this tube. And when you actually put the tube around the correct way and look at it as if you were looking at a test tube, heavier parts of what they were looking at are on the bottom and lighter parts are on the top. So actually what they did was they grew their E. coli, as I say, and obviously they needed a source of nitrogen because you need nitrogen in order to make proteins. And so what they did was they used two different forms of nitrogen. They used heavy nitrogen, or nitrogen 15, and light nitrogen, nitrogen 14. And they kind of do what it says on the tin, really. Heavy nitrogen actually is heavier, it's got a greater mass. And so if you were to spin both samples of nitrogen 14 and 15 in this centrifuge, you'll find that the 15, if you look at this tube here, will be lower down the tube because it has a higher mass. Okay, so they were using these two isotopes of DNA to prove their hypothesis, okay, because heavy DNA will be denser, as I say, and will appear in lower down in the tubes. Right, okay, so we start with this basic principle then, that the bacteria, the E. coli, are grown in an environment of nitrogen 15 or heavy nitrogen. And actually, if you have a look at this diagram here, you can see from the little tube that all of the bacteria in generation 0, so right at the start, just had nitrogen 15. So when they spun it down, there was a band, right, and they were able to show up these bands with their work. There was a band low down in the tube. And then they allowed the bacteria from then on to grow in nitrogen 14. So they stopped supplying nitrogen 15. It was still there from these starter molecules here, but they then supplied nitrogen 14. And after one generation, what they found was that they found a slightly higher band in the tube, slightly higher, okay? And then they let it grow for another generation. You can see the time down here, roughly about 20 minutes every time. And in the second generation, what they found was another band at this sort of slightly higher position and a completely new band. And on that went, so the next generation, that original band there just got smaller and smaller, and then this band above it got larger and larger in proportion. And that was explained by the diagram you can see here, because in semi-conservative replication, what's happening is that you've got the original DNA is acting as the template. So we have our template here at the top in generation 0, and what happens is that actually that is used, so this strand on the left is used to make a brand new molecule of DNA with its new DNA alongside the template, and of course the other side of the strand exactly the same. So when you look at these two generations in the tube, you can see heavy nitrogen 15 is lower down in the tube, you can see it over here, but in generation 1 you haven't got any heavy nitrogen. What you now have are DNA molecules that are containing half heavy and half light, because we've now switched to light nitrogen for the bacteria to grow. So it's not a light band, and that's really important. That's something that people get wrong with this. They think, oh, they must now be all light. No, it's not. It's an intermediate form between heavy and light. and then you let it grow again into generation 2, and you can see that the intermediate form is still there. You've still got, just like you did in generation 1, an intermediate form, but you now have bands of purely nitrogen 14, and then you let those go again, and so you see every one of these strands in generation 2 is used as a template with nitrogen 14, light nitrogen, and you can see the results there. So you can see what I'd said, that over time the light nitrogen is getting higher in its proportion, and more of it higher in the tube, and you can see there's still some heavy nitrogen there, but in proportion less and less of it as time goes on. So I think you can see there that I've said you start with a heavy isotope, and then the original DNA acts as a template, and then at the start all the bases were heavy, but then we switch to light nitrogen 14, and after that point no more heavy nitrogen 15 is available, and the proportion of it gets less every time. So let's apply that now to a question, and this is a question that when it first came out foxed quite a lot of people, because there's a lot of information here. So in 1958, Matthew Meselson-Stahl conducted a series of experiments that demonstrated that DNA replication is semi-conservative. So you can see here the position in the centrifuge tube of the DNA molecules, and you can see that the further down the tube, so this is the bottom of the tube here, the further down the tube then the heavier the nitrogen. So this is an increasing molecular mass. You've got heavy nitrogen down here, you've got light nitrogen up here. And the other thing shows the relative, it's like the proportions of the nitrogen in the bands, and you can see in generation 0 we've actually got, just because this here is the heavier mass, molecular mass, so we've just got heavy nitrogen 15. In our first generation we've got our intermediate. Remember, it's not heavy and it's not light, it's intermediate. And then this is an interesting one, because in generation 2 we've still got some of that intermediate, but we've now got the appearance of the light band. And you see again, this is why this graph is so important, because in generation 3 we have now got a higher proportion of the light. Okay, so the question is going to ask us, here we go, I think I've probably been through all of that, so I don't think I need to actually go through that again, but you know, as I say, you can go back at your leisure and look at that. So I've then said the first thing I do with these questions is say you've got to find 3 parts to the question for these 9 markers that you may come across, and in this case it asks about your own knowledge of Meselson and Stahl's experiments, to explain their experiment and the results they've got, and show how that supported semi-conservative replication, and then predict how the results would have differed if the theories had been correct. And that is, so what they're asking about there is, in the previous slide you can see the conservative and the dispersive replication, and you know, they tell you a little bit about that as well, so you don't need to know a lot about it. The focus is going to be, you know, how you relate that to the results. Okay, so moving on from that, so what we can see here is, just get rid of that a second. So you've got to put your information in there. You have to actually put the detail down, because it's saying your own knowledge. So roll your knowledge out this time with heavier nitrogen would form a band lower down, lighter nitrogen would form a band higher. Okay, it's really important for you to do that. And actually in generation 0 we've just got heavy nitrogen. Okay, so we've just got the heavy nitrogen template, and the 2 new strands in generation 1 will be intermediate, because they'll each have formed from an original heavy and the new light. Okay, so that's the intermediate. By generation 2 you've got the intermediate template has now been used to make 2 light strands as well. Okay, so you'll see one as intermediate and one as light, and by generation 3 you'll see replication will have continued. Okay, now what I suggest you do in all of these cases is you actually, again, you get your colours out and you actually draw it out for yourself, and that's when you will really understand, and that will help you as well in actually answering the last two parts to the question. Now, in an exam, obviously, or in tests, time is really short, so you know, if you need to do it to get, you know, your head around this, then do it, because that will actually sort of generate some marks for yourself. So I just wanted to say as well, the other thing while I'm at it is just to say there's plenty of images around, but be careful about the images you're looking at, because some experiments say, oh, we've got a control here, and it's just showing you where the 15 and the 14 nitrogen bands are compared to the next generations, you know, and this is another view of the sorts of images. So I'm just saying, just take advantage of all the images you will find there, okay, and relate it back to all of those bands. So now we've talked about the Meselson-Stahl. Now what we need to do is we now need to look at the dispersive and conservative methods. And if we look at dispersive, remember I said it's a bit patchy, all right, it's a bit fragmented. So in dispersive, what we'd have is we'd start with heavy nitrogen, but in the first generation, we would have all of the DNA would be a mixture of 15 and 14. So we'd have all of the 15 DNA split between all of the molecules. So that is one really important part about this, okay? So all of the DNA will look like this. There won't be a heavy, there won't be an intermediate, there won't be different strands, it'll all be this mixture. So there'll be an intermediate mass, I'm saying, but it will be getting lighter and lighter as that heavy nitrogen we started with, which is forming these fragments, gets less and less in proportion. And then the conservative method, so again, we've got to look at the results, because that's what the question's been asking us. And actually, for conservative, you see, because we always start, so let's look at this diagram here, the conservative is there, so we will have the 15 nitrogen molecule, the double-stranded there, in its entirety, and it will never be used as a template, so it will always appear. So I've said there, in conservative replication, we'd expect to see it in other bands, but on the diagrams that we saw in the exam question, we can't see that. So that is evidence that actually it is not conservative replication. There's no intermediate form of DNA at all, but we can certainly see that on the diagrams. So there we go. You know, if you follow that bit of advice, I'm hoping it's 9 out of 9. Now, I just want to, I'm just sort of coming to the end of things here really, because I just want to show you some of the things that certainly my students have found really helpful. And I'm just going to give you a little bit of a taster really, and a little bit of a reason to sort of to come to the second, third and the fourth sessions in this series about DNA and cell division, in fact topic 1.5, and that is dice sheets, because what I do with my students is that I write out, we call these, you know, dice sheets for obvious reasons, and there's not masses of detail on there, but what I've done is I've set up questions in rows here, and you'll see this is all about, you know, structure of DNA. This one's about ATP and nucleotides, and it goes through, and then we'll eventually get to protein synthesis, okay, and the triplet code. Now, every one of these questions then is answered here. So this is a template, and you can use this yourself. Now, what I will do as we get to sort of the end of the 4 sessions, by the 4th one, I'll publish all of the answers as well. So I'm just going to go back to it, because by the end of our 4 sessions, then you will have answers to all of these. And the reason my students like these, and they like us producing them in the department, which we do for every topic, is it's a way of getting people to help you with your work. So you can give them to somebody who's not a biologist. You know, your family may not have done A-level biology. Dim problem, no problem at all there, because what you can do is you can say, right, okay, then, you know, you can say, right, ask me 5 across and 3 down. What is the structure of a DNA molecule? And then once you've got the answers, somebody there then can check, and they can go over it with you and can give you feedback. So we find these really, really useful. Okay, so those are the die sheets, and as you can see, as I said, there's a topic per line really, so feel free to use these in your own revision, but by the time I get to the end of the topics within unit 1.5, as I say, the answers will be there for you. Okay, so there are lots of other things that you can do. There's a little plenary task there if you've got the time that you can go over yourself and answer the questions. The reason I picked this one is because I've shown you this diagram earlier on, and it talks there about controls. And remember, a control tube is a reference tube. So we need a control, don't we? Because we're going to need to be able to see where the heavy nitrogen is, where the band is, where the light nitrogen, the 14 band is. So it's really important that we've got a control as a reference to actually look back on when we've got our first, second, third, fourth generations. And I think the other thing that I say to my students when you have a long answer question like this, and in fact my students have done this question with me, what I say is, right, get all those keywords together, and that's why I did that with the DNA question at the start, because that's what I call a keyword stack. So you stack all your keywords together. You know, they can be in any old random order, whatever occurs to you, just get them down in a stack. Then, now people are different. They might want to do 3 before 2, up to you. 2 and 3 you can do in any order you want. But what you can then do is think, right, that keyword I'm now going to put in a sentence. And then finally, you'll put your sentences in some kind of logical order. And don't forget, when you do these QER questions in biology papers, part of the marking is that you've answered all 3 parts to the question, as we did earlier on, but also that actually your thinking is chronological. It makes sense. One point is following another. Okay, so I thought that might be something I could leave with you to have a little go. But all of the answers you have all of the answers already. This might just be an opportunity now to see something fresh and think, I can do this. I can go back through those PowerPoints and I can absolutely nail this question and get 9 out of 9. But I'm suggesting that maybe you start with writing down some keywords with sentences to actually start with as a strategy to get your full marks. So I'm coming to the end really, and so what I want to say really is thanks very much for watching. Good luck with your revision. I really hope it's helped. I really genuinely do. So there's plenty of other information out there. There's plenty of past papers. There's plenty of resources on the WJEC site, although I'm pretty certain your teachers will have got that there to show you. Don't forget the revision guides that are there as well. By the time, you know, at your age, you've actually got to the age where you know what works best for you. But I'm going to encourage you now, please, to watch my next session in this series. Now, this one is, I've called it DNA 2 protein synthesis, because this is all about the triplet code and how it is transcribed and translated into proteins. And I'll talk about all of the stages in that. I'll also talk a little about structure of the different forms of RNA as well. So can I finish again by saying thank you very much, and our next catch up is on Wednesday the 10th of March. So that's next Wednesday, and actually I've put there that it's 4:20, but in fact it is 4:30. All right, it's 4:30 until 5:10. So thank you very much and goodbye.